Thanks very much. Yeah, it's always dangerous speaking immediately after Lou because not only does he give a really good talk, but also he always insists on using his own computer, so there's always a risk of things going wrong. Thanks very much, Martin, and the other organizers for having me. For those of you who are familiar with my talks over the years, you will note two rather dramatic departures from the norm in this presentation. The first one is that my slides are not ridiculously ugly, which is largely because they were put together mainly by my amazing Director of Research, Caitlin Lewis. Is she in the room? Anyway, many of you know her. And the other thing is that I'm actually going to show you some data rather than just giving you crazy ideas. But don't get your hopes up too much because the study I'm going to be describing is still ongoing, so everything I'm going to say will be rather interim. But anyway, I'm going to describe a really big and I believe really important pioneering study on mouse lifespan and health span that I very much hope will be the beginning of a trend. In other words, that not only we, but other people will adopt this kind of study design. I will explain why that might be difficult and indeed why it hasn't been done before. And I'll talk a little bit about what has been happening. So, first of all, obviously, you know, we've been doing mouse lifespan and health span experiments in this community for an awfully long time, and we've learned quite a lot from them. But we do know that the reliability of translation of things that work in mice into the clinic is distinctly low. A lot of things work in mice and then don't work in humans. Nevertheless, a higher proportion of them work than things that don't work in mice, so mouse experiments are still useful. Now, as most of you will probably know, my work over the past quarter century has focused on a particular way of going about doing something about aging, which is, rather than trying to slow aging down, I want to try and reverse it by essentially doing preventative maintenance and repairing the various types of molecular and cellular damage that accumulate in the body. I personally believe that there are good arguments, albeit, I guess, somewhat circumstantial at this point, that those kinds of interventions will translate with higher hit rate than across species than drugs do. And another aspect of this is the combining of these things. Any damage repair approach is inherently a divide and conquer one, where we're going to end up having to do a lot of different things to the same individuals at the same time. And as Brian Kennedy mentioned earlier in the meeting, when you do that with small molecules, all bets are off. You get a lot of cases where there are going to be antagonistic interactions, things cancel out and so on. So I believe that that's also going to be less likely to happen in the case of these damage repair interventions. Really, both of those conclusions come from the fact that the target of the intervention is something that is essentially not participating in metabolism until such time as it becomes so abundant that it starts to be problematic. But the reason why we only get sick late in life is because the body's set up to tolerate a certain amount of the damage that's progressively accumulating since the whole of your life. All right, so I'm going to describe what we're doing with these studies. We call them the Robust Mouse Rejuvenation Research Program, and you'll see why that is. I'm going to talk about the overall rationale and the criteria by which we choose the components of the design, and roughly how we went about it 2 years ago, and how we are going about it for future ones. And then I'll talk about the study design itself, as well as the results. First of all, we are doing damage repair. We're doing rejuvenation, which means that it doesn't make sense to start with young mice. Of course, most studies on mouse lifespan are done starting young. We're not. We're starting at 18 months of age, in middle age. Many of you will know that there have been 1 or 2 studies recently published, very, very nice studies published by other groups, which start very late in life, like when the mice are already at their median life expectancy. And those are very valuable studies, but they're really asking a different question than studies where you start in middle age. Because, you know, if half your mice are already dead, then the mice that are not dead are close to being dead. They're definitely going downhill, which means that, in a way, you're identifying ways to extend sick span rather than lifespan, and well, rather than health span, rather. And so we want to start before the mice get sick. As I say, we're going to be combining a bunch of these things, and that's what I'm going to spend most of my time here talking about. And the biggest thing that is a real departure from almost everybody else in the field right now in terms of lifespan studies is that we're not just using drugs. We're not just using things that are orally available. We are very much using cell therapies and gene therapies as well. I'm firmly of the view that we just won't get very far, very much further than we already can, in terms of postponing the health problems of late life, whether in mice or in humans, without going in that direction. But this is really challenging, not just technically, but financially. Getting the funding to do all of this is something that is really, it's almost antithetical to the incentive structure of pretty much everybody's situation. It's not really attractive to the private sector because we're taking things that have already been developed by other people. So insofar as there's any intellectual property at all, it already exists in other people's hands. And this doesn't really generate any new intellectual property. The next thing is that from an academic perspective, it's not really the kind of thing that gets you high-profile papers in Science and Nature and Cell every 10 minutes, which is what you need to get promoted and get your next grant application funded, because it doesn't really explore mechanisms of action, or at least not in a particularly novel way. Furthermore, when we start late in life, there's a higher probability of getting negative or null results. I think this is the main reason why there is so little appetite in the community for doing late onset studies. And, I had a great time over the past decade lambasting the NIAA for not moving in the direction of late onset studies in the wake of their accidental discovery that rapamycin is quite a good life extender, even if you start late in life. And many of you will know how that happened. And the biggest thing that makes this very hard to fund is that most of the money needs to be spent at the beginning of the of the study. First of all, you've got to buy old mice. They're not cheap. But the main thing is that these studies, because they are damage repair, you basically do them once at the beginning of the experiment, and then all you have to do after that is keep the mice alive and monitor their health, which is less expensive. So I'm in the unusual position, as you all know, that I lead a non-profit that is funded pretty much entirely by philanthropy. Thank you. That means that I get out of this incentive problem. I just have to convince people that they can trust me, that I know my shit, and that I'm doing sensible things. And the other thing that it helps is that people who work for me are not looking for their next job. And therefore, I'm not worried about publication priorities and things like that. So I can do this kind of talk. I can give interim reports on things, and indeed many of you will know that I've been putting out interim reports on this study on Twitter and Facebook every month or so for the past 18 months, which is very handy. But the challenges that I'm listing here on the bottom right still must not be belittled. So what are we doing? Well, as I said, we're starting in middle age. We're starting specifically with normal mice. We're just using C57 Black 6, the way the large majority of mouse lifespan studies have been done over the years. We're not interested in doing anything with transgenic mice or accelerated aging mice or anything like that. The way we are choosing what to use in such study is initially by looking for evidence in the literature from the community for benefits in terms of both mean and maximum lifespan. We'd like to see that from more than one study, of course, ideally in both sexes. Key thing, though, it must be feasible at scale, so we're not in a position to do things like heterochronic parabiosis. However, we do want, as I mentioned earlier, to go beyond the limitations imposed by only doing orally available interventions. And one kind of side benefit that came out of work to design this study and subsequent ones was a little database that some of you may have come across, but it's only been out there on our website for a short while, so you may not have come across it. It's called RAID, the Rodent Aging Interventions Database. Essentially, every study that we've been able to find, every published study that has succeeded in extending the lifespan of mice or rats, whether transgenically or otherwise, or in the kinds of ways that might be relevant to this study. And it's quite useful. It's a very nice interface. It was generated by one of my people who has been our main literature monitor for many years. It turned out his brother was a good web designer, so we were able to get this done. It's quite nice. I recommend you have a look at it. But the study itself, well, first of all, of course, we're doing all the things that you would do for a mouse study like this. We're measuring a lot of aspects of health. I'll go into a bit more of that in a moment, but we're looking at, we're doing lots of non-invasive analyses as well as invasive ones. We're sacrificing a few mice here and there and obviously fixing and freezing tissue for subsequent analysis. Something I want to spend a little time on is the middle of this slide, the intervention groups. A large part of why this study is so big, which it turns out we're using 1,000 mice, is because it's not just a case of having a treatment group that are getting all 4 of our interventions and a control group that's getting none. We also want to have groups that are getting only one of the interventions because we want to validate that we are reproducing the results that have been published in the past and that we're doing the experiment right. And we do also want to look hard for antagonistic interactions, even though I said earlier that I feel more optimistic that these antagonistic interactions will be relatively rare in a study like this that's looking mostly at damage repair interventions. Nevertheless, it's not by any means certain. So the way we do that is by having treatment groups that have all but one of the interventions. So there you have it, 10 treatment groups in each sex, of course. And in fact, it's even more than that, because for each group except for the bottom one, we have obviously a control for the intervention that's not being given to the mice. And we do 2 different types of control. We do mock and naive. We have a version of the control where they get an inactivated version of the intervention, and that's for half of the control mice, and for the other half they get the naive. They get no intervention. And so we can tease out in that way whether the mode of delivery of the intervention is relevant to whether the thing is working or not. So yeah, it's a very complicated study. One innovation that I introduced in this study, which I, again, recommend to all of you, is the way that we do the periodic analysis during the mouse lifespan, and that includes the sacrificing of mice for tissue analysis. Most studies, pretty much all studies I've ever seen, will identify particular chronological ages at which they will do these samples at a certain number of months of age. We decided not to do that. Instead, we decided to define the points at which we would do these samplings on the basis of the percent survival. So in other words, it was group specific. So we start with 50 mice in a treatment group. We wait until 10 of them are dead. It doesn't matter when that is. When the 10th mouse dies, we kill 4 more and do this tissue analysis, and we do all the non-invasive stuff at the same time. And then we wait until another 8 have died, and so on. So this it seems to me that this has a better signal-to-noise ratio than your standard approach, because it factors out what you're measuring. If you do it the way that people normally do it, then essentially you're measuring things that you already know are correlated, because if the treatment is working, then the mice are going to be biologically younger at the same chronological age. So yeah, I feel that this gives us more information about which aspects of health are declining first in which treatment groups. So as I say, this is a whopper of an experiment. This is just a survey of how many animals we are looking at, how many necropsies, how many plasma samples, and so on and so forth. It's a lot. So let's get on to the interventions themselves. So what we decided to do this time, as I mentioned, is to use interventions that have been extremely well validated in the literature by other groups in terms of their ability to actually give life extension in mice when starting late. And we couldn't exclude rapamycin because it's definitely the one that had the most work of this sort. We might have excluded it because it's not really a damage repair intervention. I mean, maybe there are some ways in which it genuinely rejuvenates the individual, but really, you know, it's a CR mimetic more than anything else. But the other three things are very much damage repair. We first of all, doing heterochronic bone marrow transplant, we killed a lot of young mice and we purified their blood stem cells. And we injected them into the middle-aged mice after mobilization to make some, you know, to vacate some stem cell niches. This is something that's been done in a couple of studies over the past 5 or 8 years, and with positive results, so we thought, we'll have a go. Telomerase gene therapy is another example. It was quite paradoxical about 10 years ago when Maria Blasco's group showed that this could actually extend mouse lifespan. It's paradoxical because, of course, mice express a lot of telomerase already in most of their cell types. But it's really, I guess it's basically understood now that this is just because mice are so absolutely terrible at maintaining DNA maintenance that it helps anyway. So anyway, we did that. And then we used a senolytic. We decided that navitoclax was a good one to use, and we adopted the trick developed by Manuel Serrano's group of conjugating it to galactose so that it is inactivated. It's a prodrug. It's inactivated when it gets into cells that don't express beta-galactosidase very much, but then it gets cleaved off, and the drug gets activated when you don't. So this is the pipeline. We're obviously looking at lifespan, which I'll show you a bit about in a moment. And there's a lot of ways that you can slice data of this kind of multiplex design, so I'll talk about that in a second. We're measuring all the obvious things that you would do observably, you know, measuring body weight, of course, but also alopecia and kyphosis and such like. We're taking blood and doing glucose tolerance tests and so on. We're doing quite a lot of functional testing, grip strength and rotor rod for physical health, and we're doing a bunch of cognitive stuff, novel object recognition, startle response for hearing, things like that. And then, as I say, we're saving a bunch of tissues, both freezing and fixing them, for further work. And I want to actually say in relation to that, that we are really eager to collaborate with any of you on this. We do not have the expertise, the best expertise, to do all of the analyses that we think need to be done, and you guys do. You know, so anyone who has a particular specialization that, you know, is attracted by this study and would like to help, like to collaborate with us, we would be absolutely overjoyed. Please see me later. So yeah, I just thought I'd put a few pictures of mice up, but you aren't really going to be learning anything here. This is just examples of the kinds of things that we see in these studies. And so the mice on the right are getting all 4 of our treatments, and at the same age or slightly older than the ones that are not, or the ones that are only getting rapamycin. This is females. They look a good deal better. Similarly, we're looking at cases where the mice, for example, are getting 3 of the therapies, and they're doing better than the ones that are getting only 1 of them. So here's the survival curves as they currently stand. You will see that they're not complete. Indeed, in both sexes, the group that are getting all 4 of the treatments, that's the thick red line, more than a quarter of them are still alive. So we're hoping that at least some of them will stay alive for another few months. But a few of the treatment groups are now extinct, as you can see. The thick blue line is the simple control, the one that's getting no treatments. And you can see there's quite a bit of sexual dimorphism here. In the females, things look, pretty boring, really. It's kind of what you'd expect. We've got some degree of additivity of these things. You are getting the control mice doing worst and the all 4 treatments doing best. In the males, we've got some fairly crazy swapping around of the survival curves. There are, like, for example, I don't know whether I've got a pointer here. Let's assume I don't. The curve that was doing the worst early on in the experiment, the pale blue line that you can see, is now doing the third best. That's actually the group that are getting everything except telomerase, if I remember rightly. And, you know, we've got other paradoxes there. So that will take a bunch of analysis. We're not really even starting on the proper, rigorous statistical analysis of this data until the experiment is over, because there's so much to do. Obviously, there's a major multiple hypothesis issue here with 10 treatment groups in each sex. So we've got a lot to study, but we will certainly be doing that and teasing out what we can conclude with greatest statistical significance. But these are the takeaways. You know, we're doing things that nobody's done before, and I think we have shown very robustly that these experiments are delivering the promise that we felt they had. This is the kind of thing that will teach us an enormous amount about how to do actual combination therapies that matter to people in the relatively near term. We will never find that out just by doing single things. So we've got to do these experiments. But they're very hard to fund, but they shouldn't be, because even though they're expensive, they're probably, I mean, I will say not even probably, I would say they're definitely giving more information per dollar than any other experiment that anyone's doing in the field right now. We're very proud of this kind of study, and we want a lot more of them done. So stay tuned for the more interim results, the statistical analysis of everything, the analysis of all the non-invasive data that we've collected, which is now being, we're just embarking on that now. And, of course, stay tuned for the next experiment in which we're going to be doing things like, we're probably going to be doing things like epigenetic reprogramming, and we're thinking about using mouse serum albumin, which again has had quite interesting reports of benefits, things like that. But that's all mentioned on our website. So thank you for your attention. We wrote this up in an article that's just come out a couple of months ago in Expert Opinions in Therapeutic Targets, so please have a look at that. And thank you to our donors, of course, without whom this could not possibly have happened, as well as to our advisors, our collaborators, and as I said, we do want more collaborators, and also the team at the CRO with which we did this work. That's iCore Life Sciences, which is one of the most successful spin-outs from my previous foundation. They've been very, very good in making this experiment happen. And of course, if any of you are interested in helping us get the next experiment off the ground, then get that QR code on the right. Thank you very much.
Thank you. Thank you so much, Aubrey. We have time for a few questions.
Aubrey, fascinating, thank you. But could you comment on the prospects for translation? I mean, what's the end game? You do need to show, for example, at FDA, the individual components and what they contribute and so forth, so the methodology would be great. But do you see this as proprietary? It's an algorithm, it's not a therapeutic, it's a product. Is a big pharma going to pick this up? How do you see that?
Well, you're absolutely right, of course, and this is part of why it's so hard to fund these things, you know, even though it's only a few $1 million, it's not the cost of a clinical trial. But, you know, at the end of the day, we need to save lives. We need to do this somehow. As many of you know, I don't just lead scientific work. I do a great deal of public outreach and community building and so on. And a huge part of that is in order to essentially raise the attention and the understanding of what's going on in laboratories around the world, so that regulatory issues, whether driven by governments or driven by the interests of big pharma, can be modernized. So I work quite closely with people who are trying to do that, you know, with groups that are trying to reinvent regulatory structures. But absolutely, I completely agree that that kind of thing has to happen, or this will never actually reach the clinic. And I think the best way to make it to make that happen is to show that so that it does work in mice. I mean, you can call it more of a rhetorical argument than a scientific argument, but it's just as important as the science.
Yeah, thanks. Actually, a related question. You said you want to have much more studies, many more studies like that. For generalizability, what's the best choices? Should you not go to another species then, or? What's your options?
Well, so there's always a dilemma there between diversity of model versus comparability of experiments done by different people in the same model. So, I mean, the large part of the reason we use Black 6 is because so many other people have done it. We thought about using Het3. When we started this experiment, Jackson Labs just couldn't supply enough Het3. Now they can, but we're probably not going to do it anyway, because if you do, then you're talking about genetic heterogeneity. Every mouse is genetically different, so you really can't do cell therapies, at least not without immunosuppression. And we want to do cell therapies, that's for sure. So yeah, I mean, there are arguments both ways, and I would certainly love to see this kind of thing done in dogs, for example, but at this stage, where the goal is to learn as much as possible about how to combine things, what things combine well, I think the value of being able to compare different studies done in different labs as closely as possible is quite strong. And therefore, I would tend to say that, on balance, it's best to just stick with the same model.
All right. Thank you so much, Aubrey. That was a fantastic talk. Very exciting. Looking forward to the updates in the coming months and years.